My presentation is on the effects of centrifugation on the gluteal muscles during 60-day bed rest. First, I'd like to introduce that the muscles undergo degradation in space. On the left, we have a diagram of a person standing in Earth's gravity. The black muscles over here have been highlighted to indicate that they are postural muscles, and these ones are the lower limb muscles. This includes the gluteal muscles, the thigh muscles, and the calf muscles. And these are used in particular to help us stand on Earth. However, in space, the body undergoes a decreased axial loading process due to microgravity. And so all the muscles, including and especially these postural muscles, experience a deloading, which causes them to decondition and degrade. Currently, the exercise protocols on the International Space Station are still insufficient to mitigate these degradation processes. My research focuses on the degradation of the gluteal muscles. Unfortunately, there's only been one study so far that's looked at the gluteal muscles in space. I'd like to point out that the gluteal muscles have different functions. Normally, and previously in the literature, they've been more often treated as a single group of muscles performing the same function. However, recent studies have shown that they have unique functions and anatomy. For example, the gluteus maximus is the primary hip extensor and is the largest muscle in the body. The gluteus medius, in contrast, is the primary hip abductor, and the gluteus minimus is the primary hip stabilizer. Of course, these three muscles also play other roles, but these are their primary functions. They also have different functional and anatomical changes during pathology, like osteoarthritis in the hip and when people need total hip replacements. However, the effects of disuse on the individual gluteal muscles as opposed to the group itself are poorly understood and require further research. We can measure gluteal muscle deconditioning in various ways. Firstly, we can look at the muscle volume, and the shrinkage of the muscle fibers is called muscle atrophy. In this diagram on the left, we have a schematic diagram of a muscle fiber undergoing shrinkage or atrophy due to a net protein degradation process. The photomicrograph underneath is of a normal muscle fiber before spaceflight, and on the right is after spaceflight. Muscle atrophy has previously been associated with a decreased maximal force generated. A more novel way of measuring deconditioning is with intramuscular lipid concentration, or ILC. And this can be basically defined as the percentage of adipose or fat tissue in a muscle. In these MRIs by Engelken, the healthy control subjects in one study demonstrated minimal ILC, as shown by the minimal fat intensities, highlighting in white, of the gluteus muscles. However, the MRI of the patient with non-specific gluteal insufficiency shows increased ILC in the gluteus minimus and medius especially, but also the gluteus maximus here. ILC has been correlated with aging, poorer physical performance, and a sedentary lifestyle, as well as pathology. And magnetic resonance imaging, such as these examples here, can reliably measure both indicators of muscle deconditioning. Unfortunately, there are very few studies on gluteal muscle atrophy and ILC compared to other muscles, such as the back muscles or the calf muscles. Since there are very few astronauts to analyze, we need bed rest studies to increase the sample size and simulate the effects of microgravity. During these studies, the subjects will lie at 6 degrees head down tilt to mimic the fluid shifts that we see in microgravity. And during this time, there is a decreased axial loading as seen in space. We can use artificial gravity as generated by a short arm human centrifuge to generate a chosen axial or head to foot force on the body as seen on the Earth. This centrifugation has been shown to be protective of other body systems, such as the cardiovascular and neurovestibular systems. However, there's not enough to show that it's protective of the muscles. It's also contentious as to whether we use a continuous bout of exposure or intermittent bouts with breaks in between. Only one study so far by Mirkovic et al. Has looked at the individual gluteal muscles in bed rest, but they used a resistive exercise as an intervention rather than AG, and I'll be talking about that later. And no studies have looked at the effects of artificial gravity on the gluteal muscles in bed rest. That takes me to my aims and hypotheses. Firstly, we aim to evaluate the effects of 60-day head down tilt bed rest on muscle volume and ILC in the individual gluteal muscles. And we hypothesized that bed rest would induce atrophy and increased ILC in these muscles. Our second aim relates to AG. We wanted to evaluate the effects of daily AG on muscle volume and ILC in the individual gluteal muscles during 60-day bed rest. And we hypothesized that daily artificial gravity would produce a head-to-foot force that was sufficient to mitigate atrophy and increase ILC in all the gluteal muscles. So here's the study design. This study was conducted last year in Cologne, Germany, as part of the Artificial Gravity Bed Rest Study, sponsored by ESA, also known as AGBRESA. So many research groups were looking at the same cohort. We were able to recruit 24 healthy participants, 8 of whom were female, who fit these physical parameters, as well as other inclusion criteria not listed here, and ethics was approved. On day 1, the baseline data collection MRI was collected, and here's an example of one of these MRI in the transverse plane of the hip. Then the cohort underwent 60-day head down tilt bed rest. They weren't allowed to get up during the entire 60 days. They were split into 3 groups. The first group did not receive any artificial gravity at all and stayed in bed the whole time. The second group received a continuous 30-minute bouts of artificial gravity daily, and the third group received 6 5-minute bouts with 3-minute breaks in between daily. There were no differences between these groups at baseline as analysed by a one-way ANOVA. There was another MRI taken at the end of the bed rest to compare to the baseline. The statistical analysis used was a 2-way mixed model repeated ANOVA, and this is one of the strengths of the study because it is quite a robust technique. The independent measure was the group. The dependent variable was the volume and ILC. And the repeated measure was time. Note that the data, the way that we were able to extract data from these MRI was through a semi-automated MATLAB-based program in which the muscles were manually segmented, and these values were automatically quantified and pasted into an Excel document. This process was shown to be highly reliable, as shown in by the ICC value of greater than 0.935. So what did we find? The first thing we found was that bed rest induced muscle atrophy. I'd like to orientate you to these graphs, which I'll be showing in the next 2 slides. For example, this GMAX graph, in the x-axis, we have the group of intervention, and the y-axis shows the volume of the muscle. The blue is before bed rest and the orange is after bed rest. Each circle is an individual participant, but the most important part of this graph is the square, which shows the mean value. We can see for the gluteus maximus that the time interaction was significant, less than 0.05, which means that bed rest did induce muscle atrophy. However, whether you were in control group, continuous group, or intermittent group did not affect how much atrophy you got, as well as group by time. So whether you were in a certain group did not affect the rate of change over time. The same can be said for gluteus medius and gluteus minimus. So artificial gravity here did not affect muscle atrophy. Moving on to the ILC, bed rest was shown to induce fat accumulation in the GMAX and the GMed only. And this can be seen by the significant p-values for time and the increases in the means for all 3 groups of these 2 muscles. However, interestingly, the gluteus minimus did not show a change in the means of the ILC. And again, artificial gravity did not mitigate gluteal muscle ILC in this case. So what does all of this mean? Well, in terms of atrophy, there is very scarce literature to compare it with. But here are three of the most similar ones. Our study is shown on the top row here. One study by Mirkovic et al., which I told you about before, had the same cohort and demonstrated gluteal muscle atrophy for all three gluteal muscles analyzed separately. Importantly, no other study has investigated the gluteal muscles separately during bed rest, which is an important point given that the gluteal muscles have different functions. For example, a 35-day bed rest showed atrophy in the gluteal muscles together, and this is shown in this graph here. And in a 17-day space flight for 4 male astronauts, atrophy was also demonstrated in the hip region. Again, artificial gravity did not mitigate muscle atrophy. This is the first time that artificial gravity has been used as an intervention for atrophy. And we suspect that perhaps the artificial gravity was perhaps too short or too weak, thus not having an effect. However, the gluteal muscles are more accustomed to generating loads that are greater than 1G, because they're not just used for standing up in the 1G of Earth, but they are used for very complex movements like squatting and running, which bear greater loads than 1G. And so that's why they might not have been as stimulated while in the centrifuge. Again, Mirkovic et al. Showed that resistive exercise may be effective in a previous study, and so it could be possible to combine exercise with AG in the future. On to ILC now. Bed rest increased ILC in the GMAX and GM, but AG did not have an effect. In terms of bed rest, this difference between the muscles and the effect of bed rest on ILC is likely due to their different functions, with GMAX and GM being torque producers or force producers, while GMIN is a stabilizer. And so it's less affected by the decreased axial loading as seen in bed rest and spaceflight. And with artificial gravity, this was the first time artificial gravity had been looked at with regards to muscle ILC. And we can speculate that increased ILC implies a loss of contractile or functional muscle tissue, and therefore would be a very useful determinant of function, in addition to things like atrophy and other functional tests in the future. So how is this research significant? Firstly, we can use this data directly for astronauts to look at the effect of space on the gluteal muscles, and thus develop targeted exercise for long duration space missions. We can also transfer this knowledge to terrestrial patients, people with hip disorders and hip dysfunction. We can understand with the control data of this study how bed rest and sedentary lifestyles affect the gluteal muscles, and thus develop targeted exercise for hip rehab. So where do we go from here? As I said before, using other artificial gravity regimens such as longer exposures and exercise would be useful to see if this would have an effect. Also, it has been shown previously that the spatial distribution of ILC within one muscle is different. For example, we could divide the muscle medially to laterally to see if there's a difference in where the fat accumulates. And that might provide more insights into the physiology of this ILC. And finally, having greater sample size would be useful. These sorts of bed rest studies are very expensive and logistically challenging to conduct. And so performing more of these studies would be useful to help understand the effect of bed rest and AG on the muscles. And this is a significant limitation with regard to sample size. However, the data that we collected in this study may be able to contribute to a systematic review further down the road.